everybody. So my name is Toni Marco. My passport. Everybody knows me as Tony. Also, Chidi knows me as Tony.
Yeah, sorry. I keep doing that.
We know from many years ago, actually. Many years ago.
Could you stand away from the screen?
Yes, I could, but I don't want this light in my face.
That's brilliant.
Yeah, I think that's the best.
Is it better?
Yeah.
Okay. So thanks for the invitation to talk. It's nice to be back after 4 years. I think last time I spoke in a pub about science. In the pub I spoke. Today I'm going to talk about the human genome. But I'm going to talk about the human genome and how sequence technology has impacted the way we see the genome. I suppose if you are here because you are curious and you know what a genome is, but anyway, I'll tell you later what a genome is. But before talking about the human genome or a genome, I would like to talk about this gentleman here, Plato. You familiar with Plato? You study in high school, Plato, you suffer, Plato philosophy. This, whether you like it or not, I don't like it much, to be honest, but whether you like it or not, he was the most influential figure in Western philosophy. He was so influential that even science, up until 100 years ago, was done the way Plato was saying. He was very influential. Plato had many ideas, many theories, many things. One of them is what is called the theory of the forms and ideas. He proposed in this theory that the reason why you see an object or an animal, like for instance a horse, right? You see a horse, it doesn't matter if the horse is white, black, big, small. You have like all shapes, colors. Even if you have a mutilated horse, it doesn't matter. You recognize immediately it's a horse, right? So he proposed that this is because there is a real horse somewhere, not in Earth. There is a real horse, the idea of a horse, and this is the real thing. And these horses that you see in the street, the countryside, they are just projections of the real horse that you can never see, right? This is the theory of ideas. This simple idea has a tremendous impact on the way we think, on the way all Western philosophy and science was structured. And the most iconic example in biology, in my area, is that of the types of a species, or types in a species, which can be basically put like in a museum, right? Museum nowadays are the places that you bring your kids to drive the security guys crazy. But museums used to be places of science. And the reason why they're places of science is because they have specimens, they have individuals of horses, other animals, preserved. So scientists could go there, study them, and this is the type of the species. And then they go into the wild and they measure the other animals, right? And they measure these animals and basically compare them always with the type that they have, this type. This is how biologists or so-called natural historians in the past used to do biology, right? But it was thanks to Charles Darwin that we know that this concept is really not quite the case. So species are not one version and then variations across the version. Species are populations. So it's not that there is one horse, one real horse, and then everything else is something similar or dissimilar to the horse. It's that the whole pool of horses in the world, they make the population concept of a species. And that's how we work now in biology, except for the genome. The genome still, probably you've heard many times, or you read in the news about the human reference genome sequence. The human reference genome sequence is one sequence, is one. And this is this picture explains well what I mean, because this is a picture of a museum, the World Contrast Museum in London. And they have a printed copy of the human genome. So anything that is not in this copy of the human genome is a variation. A variation of what? A variation around this idea, this Platonic idea of a genome that has been sequenced already. So we want to go there. We want to know the genomes of everybody. If not of everybody, of a representative sample. We cannot just stay like in Plato over 2,000 years ago and be content with a reference genome. We need more than that. We need like a proper time genome, as I explained during the talk. Although I know you know what a genome is, I'm going to make a definition of a genome, but just for everybody to be on the same page, but from the point of view of developmental biology, or just development or function. This is a cartoon of a human embryo, right, that is developing, and something magical or scientific happened in this bit here. Is that there is a protein that is encoded in a gene called Sonic the Hedgehog, like the video game. So this protein is expressed there, and this is telling all the cells around that they have to start to express all the genes, right? So these are the genes that will be expressed, and they start to interact with each other, up to the point that all the genes called the TBXs start to be expressed in different parts of the arm. And this is going to tell the body whether it's going to be the anterior or the posterior part of the arm. All these things together interacting with this, and also with Sonic, will trigger the expression of all the genes, called the Hox genes, that will be expressed in different bits of the arm, and they will tell the identity of each part. What is going to be a finger, which finger is going to be, the ulna, etc. Right? So what I want to tell you here with this slide is that the genome, as probably you read in many places, have genes. And genes are the ones that code for the proteins that are involved in all these processes. But the genome also codes for, and actually most of the genome codes for, the regulatory interactions. So this is even more important in terms of quantity. The genome codes for the products, but also for the interactions. That's why it's important to know the genome. It's not telling you only which things are in your body. It's telling you how they interact to build the organism. So, what is the genome? How is the genome? As I said, you're here because you're curious and you know what a chromosome is. So if something is in your cells, you take the nucleus and you can get a chromosome. You don't know why, because you can't. Ideally, if you could wind the chromosome, and you will find that the nucleus, the genome, is made of something called DNA. And this DNA is a very complex molecule, actually. People think it's simple, no, it's very complex. But even though it's complex, it's made out of a complex repetition of 4 units that just for simplicity we can call TAGC. So it's the order of these 4 letters what determines not only the proteins or the gene products, but also the interaction between them. And we have around 3 billion letters in our human genome. And that's how important is the genome. If we know the letters, probably we don't know what they are doing, actually. We don't know what they do most of it. But it's the first step in order to understand how the genome can give rise to an organism. How do we sequence genome? You may think that you just take the chromosome, go to the microscope and look. Fortunately, the DNA is very, very small. So we cannot do that. We cannot just look at the nucleotides. So people had to come up with methods to sequence the genome without seeing the DNA. How? Well, this is it was not the first sequencing method, but it was the first, let's say, expandable high throughput way of status sequencing DNA. We call it now first generation sequencing. It's due to Fred Sanger, Nobel Prize. And I call it the sequencing by error. Why? Because what you do basically is to force errors in a copy of a DNA molecule. So imagine you have a tube, right? And in the small tube, you put the DNA there. You don't know the sequence. You don't know how this DNA looks like, right? And then you put all the ingredients you need for this DNA to be copied. We have the ingredients in that, right? With one exception, one difference. When you put the T, the letter, this T is faulty. And the T sometimes will work, sometimes will not work. So you just do the reaction, and the reaction is to copy the sequence. You don't see that. You know it's being copied. But it's fine. You don't need to say it. The only thing you know is that when there is a T, there will be an error. And this reaction, this copy will stop. In some files, but in some others, we keep going until it finds another T, and it will stop. And you can do this. Then, instead of a T, you use a faulty A, a faulty G, a faulty C. So if you could read this, you would be reading the DNA. Actually, what you do is you take all these reactions and you put in a big jar, like a jelly, this big. You don't do this anymore. I'm talking here about the 1970s, 1980s. You run it through an electrical current, so it separates the different reactions, and you can stain and visualize those reactions that stop. And then just by reading this, you can read the sequence. I wrote something for you to look. If you want advice, just stop me and I'll show you so you can see it in close. But these are pictures, like radiographies of sequencing gels of this time. I don't know if you can see, maybe with the light, there are like some spots, some dots. Can you see these dots? So basically these dots are in 4 lanes. These are the A, C, G, D. And basically you go, each dot is an error in one of these copies. So you just go and read A, C, C, A, T, G, like that. People used to sequence like that, looking by eye, the errors of the reactions. Obviously we don't do that anymore. We just, we have machines that read this for us and they look in smaller things. So this is the sequencing by error. That's how we sequence the human genome, actually. The human genome was something magical that we just look at the microscope and we know the letters. That's how it was sequenced. The Human Genome Project was a 15-year plan to sequence 90% of the genome. Why not 100%? Because it's pretty much, or it was with this technology, impossible. There is something called repetitive DNA that doesn't allow you to sequence, at least with this technology. So the idea was to get 90% of the sequence, and in 1990, it started with Jim Watson as a director. You may know Jim Watson was the guy with the DNA. may be brilliant, but he didn't do much in this project, so 2 years after he was kicked out. And Francis Collin took over. So in the next 6 years after Francis Collin took over, they got the first animal genome sequence. The Human Genome Project also includes sequences of other animals as models, as methods. But the following year, there was a private company, Celera, led by the CEO, Craig Venter, that they basically they promised that they could sequence the human genome even faster and better, right? So they set a race between a public consortium and a private company. The following year, they published their animal genome, although it was really a crap of a sequence, but I can tell you about that if you want after the talk. The people in the community was very pissed about this. But in the year 2001, both groups published the first draft of the human genome. It was a bit of a publicity hype because it was the big draft, but it was 80%, that was not the goal. It was 80%, but they were racing, they just wanted to publish something, right? And to release to the world that the billions spent was something. So it took 2 more years of big effort of the public consortium, including people at the Wellcome Trust here, Cambridge, Kingston, until they got up to 90% of the genome, the goal. 2 years ahead of schedule. So that was like a big thing with the human genome. Ideally, well, the goal was to take samples from males and females from different backgrounds. The reality is that because of a technical error, most of the reference genome of the public consortium came from a white male. And the DNA sequence from the Celera company was that of the CEO, Craig Venter. So it became a white male. White European male. When I say European, I mean European in origin, not necessarily European, it can be American. Actually, they were American. At that stage, entered something new, the second generation sequencing. There was a new way to sequence genome, to sequence DNA, faster and better. How it works? Okay, I'm going to try to explain in under a minute what I explained in 2 lectures to my students. So if I fail, please forgive me. So basically, we start with the same concept. We are going to copy a sequence of DNA. We're going to copy a bit of DNA. We don't know what is inside, and we put all the ingredients of the reaction, except that we put only one of the letters. But this letter also has a chemical reaction coupled to it that if this letter is copied, it will emit a beam of light. And there will be a camera that will capture this bit of light, right? So we start with a G. There is a G, so then the tube will do a bit of light. Then we put a C. We wash, we put a C. And the C is not there, so no light. T, no light. A, light. And then we start over. G, C, T, A. That way, we can get beams of light every time we're copying one of these letters, right? And we can reconstruct the whole sequence. And you'll be thinking, what for? Why is this better than the other way, right? It would be similar. Well, the difference is that the other reaction in these gels, you need like a small tube that they will like really miniaturize, capillary things, but okay, very small. But still, you need some material. Whereas here, you can fit millions of reactions in a very short space. This here is a sequencing array. Again, if anyone wants to see it after the talk, I'm happy to share. This is one of the arrays we have in one of our lowest yield machines. And in this bit of glass here, you can see only this. This is like you zoom in, and zoom in again, and each one of these spots is one of these sequencing reactions. So in one of these, you can fit between 50 and 80 million of these gels that I showed you in here. That was a massive improvement. Because pictures can be done at a very small scale. We have good cameras. And then what you have to do is to put this thing inside this machine, or other machines, similar machine, and it will do the reactions and taking pictures like this. And then a computer will give you the sequence, not of 1 or 2 or 10, but of 50, 80. So the last we submitted, we got like 300 million of those. It's now cheap, it's fast, it's better. So what is the advantage of this? Well, the advantage of this is that people started to sequence a lot of people. It was just not the reference genome and then CEO Craig Venter and then all the big hopers. Jim Watson got his genome sequence as well, so you know all the big fishes with more than the genome sequence. Now people start to sequence a lot of people, other people. And this bit here is work from my own lab. So this is actually the work of Mohab and Andrea. Andrea now works for a sequencing company, and Mohab is with a postdoc in Cambridge. So what we were interested is in variation at target sites, that means at regulatory sites of some specific genes. We study many genes. This is just one example of the many. We study many. This is one gene, it doesn't matter the name, and TAP. It's a gene that simplifying this history a lot, this so any biologist in the room, please forgive me about that. It's an oversimplification. This gene is associated with the increase in the number of size of moles and development of melanoma upon sun exposure, right? So we were working in these genes for reasons very long to explain here in a short talk. But when we started to look at the African genomes, we found that this gene was heavily regulated by two other genes. It's called microRNAs, but it doesn't matter. Just two other genes were heavily regulated, such that the sun exposure was not affecting as much the influence of MTAP in the development of melanoma, right? We know the reference genome came from a European background. So what if we quantify how often this happened in African genomes, how often this happened in European genomes, and how often it happens in different human populations? And that's what we found. This is not a scientific figure, so this is just a cartoon for the talk. I didn't want to put all these regressions and things that are very boring, but for me, they're generally boring. But that's what we found. We found that the farther you go away from the equator, where the sun exposure is lower, the farther you go at the higher latitude, north or south, the smaller, the lower the frequency of regulation of this gene. Meaning that this gene need not to be as tightly regulated, and these regulations has been lost during evolution, during migration actually. So that tells us that looking at the regulatory elements is very important in populations. So when you are sequencing genomes and you are missing the genomes of different people, you may be missing a bit about genes or about proteins, but what you're actually missing is a lot of regulatory interactions that you're not going to pick up if you only look at a reference gene. This method of sequencing and using a reference genome is called resequencing. Resequencing, you get a reference genome, like the one we have, you sequence different people, and then you count the difference between your people and the reference genome. That resembles the Platonic idea, right? That we have one idea of the horse and then variation. We have the idea, the reference genome, and then the variation. But that's not what we want. The goal now, the goal for geneticists, for genomics people, is to get to a graph genome, to a graph human genome, a pan genome. A genome that is not linear. It's not just a sequence of letters, A, T, C, T, 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 T. What we want is that you can follow different paths depending on different populations or groups. That's the goal. Actually, that would be, the reference genome would be, as I said, the horse and the Platonic idea, and this is the Darwinian population idea, the graph genome, which is our goal. So in this context, the group of Steve Sassler, what they did is to sequence 1,000 African genomes. In time, it's 2 more minutes. Okay, sequence the African pan genome, which means almost 1,000 African genomes, and they found not only that there were differences, we knew that there were differences, they found that there was 10% more DNA than in the reference genome. That's massive, that's 300 million letters more. That means that by not sequencing all the genomes, all the so-called reference genomes and non-reference genomes, we are missing a bit of DNA that is very important. So it entered the third generation of sequencing, which it has many technologies, many different ways of doing it. This is one of them, but they all have something in common. It takes a bit of DNA and reads it from head to tail, and that's amazing. So this one is called nanopore. It forces the DNA to go through a small hole in a membrane. It measures how the electrical current changes, and with that, a computer can tell you the DNA sequence. And one of the amazing things of this technology is the mean ion is that it's so small, like this small, and you can plug it into a laptop and you can sequence. And these are what our students, first-year students, are using for doing some practicals on sequencing. Using this technology, it was finally the first complete human genome sequence 20 years after the first round. This is 100%, finally, only with this technology. It couldn't be possible before. The black bits and the misses in the previous release, and now they are now done. So what is this new human reference genome? Well, it's a cell derived from a single female of non-reported origin. Well, we made some progress here. But it was from a type of mole that grows in the uterus, that is formed by the fertilization of a sperm, and then the egg loses the maternal DNA. So basically, they were sequencing sperm, an egg. And the ancestry analysis reveals it was European. So again, the reference genome that was published 6-7 weeks ago, so this is quite recent, is again that of a white European male. But not everything is bad. So there are many projects. This is particularly the Pan Genome project. So the goal now is to sequence more and more and more people, but not only sequence, but getting full assemblies of genome of more people with the idea of understand diversity and not to rely on just one reference genome of a white European male. So that's the step, going from here, in the year 2000 we got there, and now we are moving slowly through here. And it's important not only because of the scientific exercise and the understanding of variation, it's important because I was going to say in the future, but it's pretty much in the present, medical treatment will depend on the genes you have, and only by sequencing your genome. Most people will know. As maybe Greg say something later. So with that, I would like to quickly thank you to the people that put the money, that was important, but even more the people that did the work, and I'm happy with all the bits I told you, and currently in my lab we have Igor, Toni, and Anna, they are also working with Greg, and to you for being here. Thank you very much.
Yeah. So, does anybody have any question for Toni? No?
Crystal clear? Or not?
I have like two different questions already, so I can. No? Well, I can just start. So, first question for me is like kind of a philosophical one. So, how many genomes you actually need? Like, if you want to know this essence of what is the horse or the essence of what is a human, how many genomes, how many human genomes or horse genomes you want to actually separate?
Yeah, it's a good question. It depends for what. For the assembly, I think the initial plans for the pangenome is 1,000 genomes. For the assembly, to have full assembly. But for diversity, there are many more. Actually, we have the Genomics England that is sponsoring this, is having the 100,000 genome project, which they are not 100,000 genome assemblies. This is DNA from 100,000 people that then they are mapped against the assembly. So for variation, we need more than that. Actually, there are more than that. A million, maybe more. We don't know. But for full assembly, currently it's 1,000, but we probably more.
How long does it take to sequence one genome now with the third generation sequencing, and how much does it cost?
Okay, good question. I don't know about third generation sequencing, how much it costs, because I know the cost of the linear, but the linear is very expensive if you want to run a whole genome. So you have to rely on something called back value, and I don't have an idea of the cost in my mind now, how much it costs. But I can tell you second generation sequencing with using a reference genome, second generation sequencing is about £250, and you get it in a week. I got mine done. So.
Will we ever get to the point where, as soon as you're born, you're getting sequenced?
I suppose it's an ethical issue. And not technical.
Are there any biosecurity concerns with releasing the genomes of certain populations?
Yeah, well.
So are there any controls over who we sequence, what data we release?
Yes. As a matter of fact, every time we have to access some databases with information from individuals, you have to get the university team to communicate with them and sign a lot of papers and be sure that the data is anonymized. But obviously, if it's fully anonymized, you cannot do anything, because if you don't know the ethnicity, you don't know the sex. So you have to be very careful which information you need, and they release only the information exact that you need for your project that has been justified. So yeah, there's a tight control. There are always ways of de-anonymizing, particularly when you get the DNA, right? DNA is unique. But at least on the paper, there are lots of controls to analyze this data. It's not something you can just download and analyze. Yeah?
I've got a question. You just told that guy that the aim is to have 1 million genomes. How are you going to store all that data?
So on the one hand, that's a big problem. But we are lucky enough, and I shared my path for many years with an astronomer. We are lucky enough that an astronomer had this problem like some 30, 40 years ago. So they are really solving this problem for us.
Okay.
So basically, because the images they get from the telescopes, these are gigabytes of data per second. So they have to manage the storage the system and so on. So we are a bit behind, but it's fine because we can learn from them. But there is a lot of people working on methods to storing genome information, like compression algorithms, but then fast compression reading algorithms. And actually, the algorithm we use for mapping to find this resequencing, they use a compression algorithm. The key bit is that you compress the genome, you compress your reads that you want to map, and you never uncompress. You work with compressed version. It's very clever algorithm. So the world is willing to transform. But many people should work with that.
Sorry, that 10%
there are quite a lot of long non-coding RNAs, and there are coding regions as well, but it is not clear whether they have like a proper function. But as I said, so the big chance that they sequence, they have some genes, but maybe they don't work as in the other genes because the other has been heavily studied. But there are small bits around the genome that are means like gaps in the genome, and they do contain regulatory elements, and these are the important ones.
Thank you very much.
Thank you very much.
Of course, we will be around for